Good morning, everyone. Good to see you. If you would, please go ahead and type your name into chat or your nickname. Thank you. Good to see so many of you here on Monday morning. I hope everybody had a nice weekend. So we're into the last week of March. Hard to believe. Yeah, we're still missing a few people, but we'll go ahead and get started. So this week we are moving ahead from the discussion of carbohydrates and carbohydrate catabolism or breakdown for the production of energy. And we're going to instead talk about protein and protein catabolism. So our exercises this week are broken into two. The first protein catabolism exercise is about the way that microbes use protein as a source of amino acids. And in the second laboratory exercise, which we'll get to later in the week, we'll talk about how microbes can break down amino acids as a source of carbon. So there are really two reasons that microbes catabolize or break apart proteins. The first reason, and the most common reason, is just to get the amino acids out of the proteins. That's the same reason that you and I break down proteins most of the time. Most of the protein that we eat is simply a source of amino acids to build other proteins. Because remember, that's all proteins are. They are a long, long chain of amino acids that are then folded up into some three-dimensional structure that's going to serve some function in our bodies. And microbes are the same. They break down protein to get those amino acids so they can build more protein, essentially. So remember, there are about 20 amino acids. There are actually more than that, but there are 20 what we call common amino acids, the ones that occur in abundance in nature. And about 8 out of those 20, are what are called essential amino acids for humans. And what that means is we don't have the ability to make them from scratch. We have to get them in our food. So we have to consume proteins that have those essential amino acids, those 8 amino acids in them. And then we break apart those proteins to get those essential amino acids, and then we can use them in our proteins. The other 12, the ones that aren't essential to us, the ones that we can build from scratch, those we get by building them often from carbon that's been harvested from other molecules. So we're going to start off today talking about this process of catabolizing protein to get at the amino acids so that the amino acids can be reused in microbial cells. Thank you, Christina. Thanks for letting me know. All right. So as usual, before I jump into our lab today, I want to ask first if anybody has any questions about anything that you've been watching in terms of lecture topics, any of the quiz questions you may be taking in our lecture quizzes, any questions at all about any of the material we've been covering. Feel free to type questions into the chat and we can talk about them. The other thing I'm going to do just very briefly is pull up our schedule for this week and looking ahead to next week, just so we can know what's coming up. So what you should be seeing right now is the course content page for the course on Blackboard. This is our to-do list for this week. We have 2 lecture topics this week about the DNA molecule. So we'll be talking about genetics in the sense that we're talking about what microbial cells, in fact, what all cells do with the DNA molecule. And remember, there are 2 things that cells use DNA for. The first thing is to make more DNA. That's pretty easy to remember. And that process where DNA is used to make more DNA is called DNA replication. And of course, as we've discussed in here, when we talked about ultraviolet radiation, sometimes DNA replication goes wrong and we get mutations. So that's also part of that lecture. The second way that cells, all cells really, including microbial cells, use the DNA molecule is to build protein. Remember that what we call genes in DNA are simply recipes, they're instructions for how to take a sequence of nucleotide bases and build a protein. So transcription and translation are the 2 separate processes that cells use to build protein from DNA. So there'll be a lecture quiz associated with each one of those lectures. And again, in lab, we'll be talking about protein catabolism. And don't forget, as usual, you have lab homework this week that's due by Sunday at midnight. I'm also going to pull up our syllabus just for a minute so we can take a look at some things that are coming up next week and beyond. We are coming to the end of March this week, so moving into April, we're really in the last portion, the last quarter essentially of the course. So I just want to remind everybody that you have your 4th lecture exam coming up next week. And in laboratory next week, we'll be talking about our last biochemical test, which is a test or several tests that are involved in respiration, cellular respiration. The other thing that's going to happen next week in lab is you're going to get your final laboratory assignment, which is the assignment where you'll be given an unknown microbe to identify. Now, you won't be conducting the tests, but I will give you the test results. So I'm going to give you a list of all of the results of tests that you've learned about this semester. And it's going to be your job to use those lab results to identify this microbe that you've been assigned. So we'll distribute the data next week. We'll talk about these tools that we use called dichotomous keys that help us in our identification. That'll be coming up next week. I do also want to just point out that you have your final lab practical exam the following week, so the week of April 12th. It doesn't mean that we don't have lab after that, we do. But in terms of all of the things we've been learning so far about bacteria and biochemical testing, those will be covered in that final lab practical exam. So just something to know is coming in the next couple of weeks as you continue to work through the labs, as we continue to work through the labs. I also just want to remind you about your pathogen assignment. Don't forget that. Hopefully you've started working on that. I've heard from a couple of students with questions about it, so that's good. I'm glad to see you've been working on it. All right. So I'm going to go ahead and move into our lab exercise for the day, which is protein catabolism part 1. And that's what you should be seeing on your screen right now, the title slide for this lab. Our objectives for this lab are to understand the most common reasons that bacteria catabolize protein. Remember, when we use that word catabolize, that's a very general term that we apply to metabolic processes that break down molecules into their smaller component parts. Remember that a molecule is just a big group of atoms that are all bound together with covalent bonds. And sometimes the cell will break apart those covalent bonds in order to get at those atoms or some product inside of that molecule. We're going to look at 2 specific tests in this exercise. One is called the Gelatin hydrolysis test. We'll talk about this one in terms of what the media is and what it's made out of, how we perform it, and how we interpret it. And then we'll also talk about a second test, very common test, called the Urease test. We'll talk about what urea is, what the urease test media is made out of, and again, how to perform it and how to interpret it. So let's begin by just reviewing some of the terminology around protein. As discussed in the lectures, there are some terms that are used when we talk about proteins that are not automatically known to all of us. This term protein means something very specific in science. It's a particular form of protein. And I'm guilty of sometimes using the word protein when I ought to be using the term polypeptide or even peptide. So we need to know the definitions of these terms and scientifically what they refer to. All a peptide is a relatively short chain of amino acids. So in lecture, you've learned about what an amino acid is, what it's built like, what the component parts of an amino acid are. You know that there are 20 of them that occur in nature. When we join them one to the other, if it's a relatively short chain of them, it's called a peptide. Generally, peptides contain somewhere in the neighborhood of tens to hundreds of amino acids. Now, that may not seem like a short chain to you, but compared to a polypeptide, that is a short chain. A polypeptide then is a longer chain of amino acids, and essentially the first form, or what we often call the primary structure of a protein. It's a long chain of amino acids, generally in the area of hundreds to thousands of amino acids. Again, all joined together with covalent bonds. And remember, we call the covalent bonds that join the amino acids in a polypeptide or a protein, we call those bonds peptide bonds. It's a covalent bond that just happens to occur between 2 amino acids, so it gets a special name. A protein then is essentially a polypeptide that has been processed and folded into a functional 3-dimensional shape. One of the most amazing things about proteins, because there are so many of them in nature, one of the most amazing things is that every protein is different. Every protein has a different sequence of the amino acids inside of it, and it therefore folds slightly differently. Some proteins look very similar. They might share, say, 70 or 80% of the amino acids in the same location. But because the sequence is unique for every protein, the sequence, the order that the amino acids are in is unique for every protein. The way every protein folds up is also unique. And that's what allows individual proteins to do different jobs. The other important thing to remember this week, especially as we talk about mutations in lecture, is that if you alter the sequence or the order of the amino acids, you're going to alter the way the polypeptide folds up. And if you alter the way it folds up, you're going to alter its function. So let's jump back in. Short chain of amino acids is a peptide. Long chain of amino acids is a polypeptide. Processed and folded polypeptide is a protein. And every protein has its own unique sequence of amino acids and its own unique three-dimensional shape. So when a microbial cell consumes protein, takes in protein to break it apart, the most common reason that it's going to do that is to get at those amino acids. The most common reason is to get the amino acids so that those amino acids can then be turned around and used to build a new protein. So they catabolize one protein in order to build, which is anabolic, to build new protein. This is the same reason, most common reason, that we break apart proteins. Most of the proteins that you consume are going to be broken into their amino acids so that your cells can build new protein. It is also possible, though, for microbial cells to break apart those amino acids. In other words, to break the protein first into its amino acids and then further break down the amino acids. We're going to talk about that process later in the week. But today we'll focus on that first most common thing that microbial cells do with proteins, which is catabolize them into amino acids. So I mentioned that we're going to be looking at a test called the gelatin hydrolysis test. And like the name suggests, this is a test that uses a particular type of protein called gelatin. We're going to examine the ability of various bacteria to catabolize or break down gelatin and release its amino acids. Gelatin, if you're not familiar with it, is just it's a very common protein. It's found in connective tissue in humans and other types of vertebrate animals, so it's very abundant in the body. We also use gelatin, right, as a dietary or a nutritional food. If you've ever had Jell-O, or you've ever had anything that has a gelatinous texture to it, gummy bears and things like that. Those often have gelatin inside of them. There is a whole family of enzymes that can break gelatin apart. Now, in microbes, these are, again, exoenzymes. Remember, we defined that term as meaning an enzyme that the microbe is going to secrete, it's going to exit the cell into the environment. Remember when we talked about the starch hydrolysis test, we said that some microbes have the ability to secrete amylase into their environment and break down any starch that might be around them to get at the glucose. Well, there's a whole family of gelatinases that microbes can, again, secrete as exoenzymes. And those gelatinases are able to break down any gelatin that might be around them into its amino acids. So the gelatin hydrolysis test is going to test for the presence of these types of enzymes, gelatinases. The other test that we're going to examine is this test called the urease test. This is a test that actually looks at the ability of some bacteria to catabolize not protein, but a waste product of protein catabolism. And that molecule is called urea. So in other words, when cells catabolize proteins, a waste product is generated. And that waste product is called urea. What this urease test looks for is the ability of the microbial cell to then take that urea waste and break it down further. This enzyme is called urease. This is another exoenzyme. So the microbial cell secretes it, and the urease breaks apart the urea into ammonia. Let's talk for a minute about the gelatin hydrolysis medium. The medium that we use for this test is based upon this protein called gelatin. Basically, what gelatin hydrolysis medium, or what's sometimes called nutrient gelatin medium, is a modification of a nutrient broth. Now, as you can see by looking at the very short list of ingredients, this is an unusual medium. This is not what we would call a growth medium. In other words, this is not one of the basal or the complex media that we looked at, whose purpose it is to grow microbes in the laboratory for us. This is a medium that will keep microbes alive for us, but it is not intended to support them nutritionally to the point where they're going to grow and divide for us like they would in other media. It'll keep them alive, and if they have the ability to secrete gelatinase enzymes and break down the gelatin, they will. So this medium contains gelatin in its full and intact form. It also contains a digest of gelatin. We saw something like this when we talked about peptone being in other culture media. Remember, peptone is a partial digest of various proteins. So peptone is a very generic term that refers to sort of bits and pieces of protein. This medium, this gelatin hydrolysis medium, also has a digested bit of gelatin in it, partially digested. The third ingredient is this mysterious ingredient called beef extract. Remember, meats and fish are some of the materials that we sometimes add into complex or chemically undefined growth media in order to give fastidious organisms a whole range of nutritional molecules that they may require. The beef extract is not being added in here as a complex growth ingredient. Instead, it's being added in because beef is a mixture of both protein and carbohydrate and fat. So again, it's going to keep the microbe alive long enough for us to run this test, but it's not going to support it dividing and creating new cells the way a growth medium would. Now, the other thing that makes this medium quite interesting is the fact that gelatin is actually a liquid above a temperature of about 25 degrees Celsius. If you're not familiar, Room temperature is right around 22 degrees Celsius. So gelatin is going to liquefy at a relatively low temperature. That means we really can't incubate these tubes the way we would with other cultures. We're not going to put them into an incubator at 35 to 37 degrees centigrade because the gelatin will just melt in there. Instead, we're going to place these tubes in a protected place at room temperature. Remember, you can simply shut off an incubator and put tubes into the incubator so that they're safe and protected and in the dark, but they're kept at room temperature. You can keep cells in nutrient gelatin medium up to about 7 days at room temperature. Some microbes take longer to break down gelatin than others. So this test can take as long as 7 days to complete. The key thing to remember is this. If the microbe that we place into that nutrient gelatin medium, if it's capable of secreting gelatinase and breaking that gelatin down into its amino acids, that medium is going to liquefy. It's going to become a liquid because the gelatin, the part of the medium that made it solid, is going to be gone. It's going to be digested away by those gelatinase enzymes. So we look at those tubes that are incubating at room temperature. We look at those tubes that have solid gelatin in them, and we watch to see if that medium becomes a liquid. If it's a liquid at room temperature, it tells us the gelatin has been hydrolyzed. It's been broken apart or catabolized. If it's a solid at room temperature, it tells us that the gelatin has not been broken down and the microbe simply does not have the ability to produce and secrete these enzymes called gelatinases. On this slide, you're looking at the gelatin hydrolysis test. You're looking at 2 tubes that have this nutrient gelatin medium in them. I want you to look at tube B here because this is what the gelatin medium looks like before it's inoculated. As you can see, as this tube is tilted, the medium is solid. It's staying put in the tube where we placed it. Now, again, we really wouldn't consider this a growth medium. It's not a growth medium. This is a test. This is a test for a very particular type of enzyme called gelatinase. And when the organism has the ability to make and secrete gelatinase, this is going to happen. The solid gelatin is going to be broken apart and the medium is going to become a liquid. And when you tilt that tube, you're going to see the liquid run up the side of the tube. So after it's been inoculated and incubated on the counter at room temperature, upwards of 7 days, if the medium remains solid, you know that is a negative gelatin hydrolysis test. If the medium turns into a liquid, you've got a positive gelatin hydrolysis test. Let's switch gears here and talk about the urease test. Again, first we'll talk about the medium itself, and then we'll talk about how to interpret the results. Now, unlike the nutrient gelatin medium, this medium is actually a basal medium. So this is a medium that can support the growth and division of cells. It has a very simple ingredient list, but it's a list of ingredients that is complete. In other words, it's got a carbohydrate in it, dextrose, that the microbes can use to make ATP. It's got peptone in it. Remember, that's a partial digestive protein. It's got things like sodium chloride, it's got potassium and phosphate in it. It's got agar in it, which is going to make it a solid medium. And it's got this indicator dye that we've seen before called phenol red. So we're going to be looking for a color change in this test. Finally, you can see that this medium contains urea. Now, it's important to remember that urea is not a nutrient. Urea is a waste product that cells do not have a use for. It gets produced as proteins get catabolized or broken down. If the microbe has the ability to break down urea with the enzyme called urease, a molecule called ammonia is going to be produced, and ammonia is going to turn the culture medium alkaline. It's going to raise the pH above 7 and turn it alkaline. That's going to cause the phenol red to turn color. Now, urease medium can be used as a solid or a broth. In my experience, most people use the solid form of this media, and it will be used as a slant tube. It is possible to mix this stuff up as a broth, but you have to use a slightly different recipe than what I showed you a moment ago. Another interesting thing about this medium is that the medium itself is slightly acidic. And since it has phenol red in it, and we've seen phenol red in other tests, the medium is going to be sort of a yellow color to start with before we even inoculate. Remember, phenol red starts off red and turns yellow in the presence of acid in a low pH. We saw it used, for example, in the carbohydrate fermentation medium. And when the organism fermented and produced an acid product, the tube turned yellow. Well, in the case of urease medium, the tube starts yellow because the medium itself is acidic. So this time, we're looking for a change of an alkaline pH. This time, we're looking for the phenol red to change color when the pH goes above 7. And the color we're going to see in those conditions is a very bright pink or a fuchsia. Now, unlike the gelatin hydrolysis test, the urease test is incubated as usual at 35 to 37 degrees Celsius. And we're going to incubate it for about 24 hours. But here's something else that's unusual about this test. It turns out that some microbes that secrete urease have the ability to break down urea very quickly. In about 6 hours or so, the test tube is going to turn, the medium in the test tube is going to turn fuchsia. Other microbes are able to break down urea, but at a much slower pace. It's helpful for us to know if the tube turns pink, when it turns pink. In other words, does it turn pink quickly after only about 6 hours, or does it take 24 hours for it to turn pink? Because that can help us identify which microbe is growing inside of it. So we're going to want to check our urease test medium at intervals. In other words, we're not going to put it in the incubator and just leave it for 24 hours before we look at it again. We're going to look at it at 6 hours and 12 hours and so on until we get to 24 hours. On this slide, you're looking at a pair of urea test tubes on the left that contain the solid form of the medium in a slant. And over here on the right, you can see the urease test medium as a broth. If you look on the far left here at this slant, this is what the medium looks like before it's inoculated. Remember, it's got phenol red in it, and it's sort of this yellowish, orangish color to start with. Normally, we would expect a medium that has phenol red in it to start as a red color, but because the medium itself is slightly acidic, the medium begins as this yellowy color. We're going to inoculate this tube by streaking cells down the slanted surface of the agar. And then we're going to put it into the incubator and check it at intervals, approximately every 6 hours. We're looking for this. We're looking for the medium to turn this very bright fuchsia or pink color. This is an indication that the urea has been broken down with the urease enzyme to produce ammonia. The ammonia raises the pH of the medium in the tube and causes it to turn this color. You're going to see the same type of color developing if you use the broth form of this medium. It starts off a yellow-orange color. A positive test then is a pink or fuchsia color change. A negative test would be if the medium stayed the same color. I want to take a minute and talk about a bacterium that we associate with the urease test called Helicobacter pylori. Helicobacter pylori is a human pathogen that infects the gastrointestinal system. In fact, this is the organism that in recent years has been discovered to be the cause of many gastrointestinal ulcers. So when a person is now discovered to have an ulcer in their gastrointestinal tract, sometimes they will have a biopsy taken of that ulcerated area looking for this organism. So take a look at this bacterium in this digital image here. You can see that it's got a rod shape. It's kind of a bent, twisty rod, so you would be forgiven if you called this a spirilla shape. Scientists are still fighting with each other over whether this is a, quote, curved rod or a spirillum. The thing to know is that it is elongated like a rod shape, but it's got a little bit of twist in it, or a little bit of a helical twist, almost like other organisms that you've seen. Notice that it has flagella at one end. It has multiple flagella at one end. That's another distinguishing characteristic of H. pylori. On the upper left-hand corner here, this is a scanning electron micrograph image showing these H. pylori bacteria in the intestinal tract. See them there? This is an image taken with a light microscope. You can see one of these cells right here inside the lumen or the cavity of a stomach gland. And then over here, this is just a lower powered image of H. pylori showing you that it's gram-negative. So Helicobacter pylori is a pathogen, and it causes a significant problem in humans, which is gastrointestinal ulceration. When a person has a biopsy done in order to look for this organism, that biopsied tissue sample will be sent to the microbiology lab so that it can be used to inoculate urease test medium. And the nice thing about this organism is that not only is it urease positive, but it is one of the microbes that is rapidly urease positive. So we can inoculate the culture medium and incubate it for 6 hours, and we should be able to see that fuchsia color developing. So the physician can get an answer very quickly about whether or not H. pylori is probably the cause of that patient's ulcers, and the patient can be put on the appropriate antibiotic. Now, as we've said before in here, we wouldn't want to stop our investigation simply at a positive urease test at 6 hours. We would want to keep investigating this organism in the microbiology lab because, first of all, H. pylori is not the only microbe that is rapidly urease positive. In fact, it turns out that there are yeast that can sometimes grow in the human gastrointestinal tract and cause similar signs and symptoms. Remember, in a previous lab, we talked about a genus of yeast called Candida. Well, there are two species of Candida that are very common in humans. One is called Candida albicans, and one is called Candida neoformans. The Candida neoformans, particularly, has to be distinguished from Helicobacter pylori because Candida neoformans is also rapidly urease positive. In other words, I might have H. pylori in that test tube, but I might also have Candida neoformans in that test tube. I have to do further tests in the microbiology lab to learn whether or not I have this bacterium, Helicobacter pylori, or this yeast, Candida neoformans. I have to do some other tests like we've talked about. I have to make a smear from that biopsy. I have to make a smear, I have to stain it, I have to look at it under the microscope. That's going to help me know which one of those two organisms is more likely to be in that tube. Now, I do want to mention that Candida albicans can also cause disease in humans. Candida albicans, we will be able to distinguish from Candida neoformans because Candida albicans is not rapidly urease positive. In fact, it is urease negative. So if I inoculate that test tube, that urease test, and I put it in the incubator and I don't get a rapid color change, and I don't even get a color change after 24 hours, I might have a Candida in there that is like Candida albicans, not urease positive. You can't stop, in other words, with just that one test result. I'm going to report it to the physician, and the physician is probably going to treat that patient with antibiotics, but then I'm going to finish my other tests and report back to the physician in a day or two whether or not we really have Helicobacter pylori. Now we're going to move on to our experiment for the day. We're going to be using 5 different organisms, and you've seen most of these before. We're going to use Staphylococcus aureus and Staphylococcus epidermidis, which are both gram-positive cocci. We're also going to use 3 gram-negative rods, E. coli, Klebsiella pneumoniae, and one you haven't seen before, which is Proteus mirabilis. So we've got 2 gram-positive cocci that we know are sometimes found on the skin of humans, as well as in the nose and the pharynx or the back of the throat. We know that humans can carry these organisms and have no problem at all because they're just part of the microbiome. But there are also pathogenic strains, and certain species tend to be more pathogenic than others. Among the gram-negative rods, we know that E. coli is a coliform. Remember, that means it lives in the intestine and it can ferment lactose. Klebsiella pneumoniae is also an intestinal coliform. Again, we haven't seen Proteus mirabilis yet in our exercises, but I'll speak a little bit more about that organism in just a moment. For the first part of the exercise, we're going to use the Staphylococcus aureus and the Staphylococcus epidermidis. We're going to inoculate 2 nutrient gelatin tubes with these organisms. We're going to place those inoculated tubes into an incubator that's been turned off, and we're going to let them incubate at room temperature for up to 7 days and look for a change in that gelatin medium. We're going to look for a liquefaction of the medium at room temp, which will tell us whether or not gelatin hydrolysis has occurred. And here are our results. Notice that the 2 tubes are being tilted. This is how you evaluate this test. You take these tubes out of the incubator and you have to tilt them in order to see whether or not the test medium has liquefied. The Staphylococcus aureus culture has caused the gelatin in the test medium to be degraded. The Staphylococcus epidermidis tube still has solid gelatin in it. So we know now that Staphylococcus aureus has the ability to hydrolyze gelatin. In other words, it can secrete these gelatinase enzymes into the medium and break apart the gelatin into its individual amino acids. So if I were to take a little bit of this test medium out, there would be no gelatin in here, but I would find a lot of amino acids in the tube. The Staphylococcus epidermidis has not turned the gelatin into liquid. That tells me it has not broken it down. The gelatin is still intact, and the medium is still solid at room temperature. The remaining organisms, the E. coli, the Klebsiella pneumoniae, and the Proteus mirabilis, were used to inoculate urease test media. Now, what you're looking at on this slide are those 3 organisms in their tubes, as well as a control tube. Remember, you always, 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 always want to run an uninoculated tube through your experiment. In other words, I'm going to inoculate with Proteus, I'm going to inoculate with Klebsiella, I'm going to inoculate with E. coli, and then I'm going to take a 4th tube that I don't inoculate, and I'm going to do everything to this control tube that I do to these other tubes. If I'm going to place these tubes into the incubator, I'm going to place this tube into the incubator as well. If I'm going to read these tubes at 24 hours, I'm going to read this tube at 24 hours as well. It's a comparison. It's a control. If I pulled this uninoculated tube out of the incubator, let's say it's 6 hours later, and this medium turned pink, I would have a problem, wouldn't I? I didn't put any cells on here. That would tell me that there's something wrong with the culture medium, that the culture medium can't be trusted, because I got an alkaline pH change without the urea being broken down. If there are no cells on here, that color change would not have come from the urea being broken down, but from some other problem with the medium. So you always have to run a control. The control is also going to be useful for comparison purposes. Now, look at the results we got. These tubes were inoculated with the 3 different test bacterium that are left in our exercise today. They were placed in the incubator and they were checked after 6 hours, after 8 hours, after 10 hours, and so on. The results that you're seeing were all achieved within 6 to 10 hours. So Proteus mirabilis at about 6 hours turned the entire tube this bright pink or fuchsia color. This is a positive urease test. If I were to interpret this, I would say that Proteus mirabilis has secreted the urease enzyme into the culture media, and that urease enzyme broke down the urea that was present in the medium and turned it into ammonia. Ammonia has a high pH, greater than 7, so the phenol red turned the medium this fuchsia color. Proteus mirabilis is known for being rapidly urease positive. Now look at the second tube. This was the tube that was inoculated with Klebsiella pneumoniae. At 6 to 10 hours, this is the result. Remember, we spread the cells right here on the slanted surface of the agar. And notice that that's where this media has started to turn pink, but the bottom part of the tube is not yet pink. We've seen something like this before. We've seen a tube that was half changed and half the same. We're going to interpret it a little bit differently, though, for the urease test. I'm going to say that Klebsiella pneumoniae is urease positive. It is. It obviously has broken down the urea to make ammonia. The pH has gone up and the medium has turned pink in this area. But the whole tube has not turned pink yet. And that's because Klebsiella pneumoniae is not rapidly urease positive. It has urease, it's going to secrete urease, but it doesn't do it nearly as rapidly as Proteus does. So Klebsiella pneumoniae is urease positive, but it is not rapidly urease positive. If I put this tube back into the incubator and came back at 18 hours or 24 hours, I would likely find the whole tube had turned pink. E. coli is my negative organism. Even after 24 hours, E. coli will not show a color change on the urease test. You'll notice that growing here, the E. coli and also the Klebsiella here, the medium has changed color, hasn't it? Just a little bit. The medium started off more of an orange color and it's turned into more of a yellow color. Remember, there's phenol red in here. That just tells me that these old E. coli cells, as they're growing, are secreting something acidic into this medium. They're metabolizing. Remember, there's dextrose in this medium. They're going to use that dextrose to make ATP. So it's not a surprise that they're producing an acid and the medium is getting more yellow over time. But this test isn't about an acid. This test is about the breakdown of urea. This is about an alkaline change. And E. coli just hasn't done that. It hasn't broken down the urea into ammonia, so we don't get that pink color change. That tells me that E. coli just doesn't have the ability to make and secrete urease. All right. So when we get together next time, hopefully the power will stay on for us and we'll have an opportunity to talk about more tests associated with protein catabolism. When we get together next time, instead of talking about the way microbes break down proteins into amino acids, we're going to talk about how we can test for the ability of microbial cells to take an amino acid and break it down. Break it down in order to get at the carbon that's inside that amino acid so we can use the carbon to build other molecules.